Hello everybody, welcome to Ademedi's channel, and today we'll be talking about a topic which is a continuation of one of the topics we've talked about in the past. And the topic I'm talking about is what we call meiosis. Meiosis. So when you're talking about meiosis, what exactly do we mean by meiosis? For people who have been faithful subscribers in the past, the first topic we discussed was mitosis, which is a part of cell cycle. Meiosis is the other part of the cell cycle now. When we're talking about meiosis is a form of cell division which involves a singular cell divided into four daughter cells. What am I saying now? That is, if you remember when we were talking about mitosis, we said a singular cell will divide into two daughter cells. But for the case of meiosis, the reverse is the case. It will be times 2 of what happened during mitosis. And also, one major difference between meiosis and mitosis is the fact that meiosis results in the formation of a daughter cell which is haploid. That is, it has haploid number of chromosomes, unlike mitosis which has a diploid number of chromosomes. Another important thing you need to consider, which serves as a major difference between meiosis and mitosis, is the fact that meiosis occurs in sex cell. By sex cell, I'm talking about the sperm cell and the egg cell, which results in the formation of a new zygote. But for the case of mitosis, that one is basically for somatic cell, that is every other cell of the body. So non-somatic cell is what meiosis actually is involved in. Meiosis is there to make sure there is continuity of life so that when there is fusion of male and female gametes, a new offspring will be reproduced. So under this meiosis, we'll be considering some certain steps. The first one is what we call phase 1. I want you to understand something before I write anything. When we're talking about meiosis, we have two major phases. We call it meiosis 1 and we call it meiosis 2. So just like we have for mitosis, if you remember, under mitosis, we have prophase, metaphase, anaphase, and telophase. The same thing applies here, just that we'll be adding 1 and 2 for each of these phases. For meiosis now, talking about meiosis 1, let's say this is meiosis 1. We'll be having prophase 1, metaphase 1, anaphase 1, and telophase 1. For meiosis 2, we'll be having prophase 2, metaphase 2, anaphase 2, and telophase 2. I will be breaking these phases down one by one for you. But before we move on, I want you to have something at the back of your mind. When we are talking about cell cycle in general, in the previous video, I made mention of something. Cell cycle consists of 3 major phases. Interphase, we have the M phase, and we have the cytokinesis. Interphase, M phase, and cytokinesis. The interphase stage precedes all these stages. It is what happens before all these stages occur. So the meiosis phase, the mitosis phase, that is the phase where cell division purpose takes place. And the cytokinesis phase, that is the phase where there will be division of the cell to form 2 daughter cells for the case of mitosis. So I want to have something at the back of your mind, which is the fact that interphase comes before all these phases. So interphase, which is the resting stage for the cell. At that particular stage, that is where the cell will begin to prepare for the process of cell division. Having known that now, let's take a look at each of these one by one. Let's start with the prophase 1 now. Prophase 1 is the major phase which differentiates mitosis from meiosis. Okay, what am I saying now? You know, I said something earlier. I said one of the major differences between mitosis and meiosis is the fact that meiosis will produce haploid number of chromosomes, whereas mitosis will produce diploid number of chromosomes. So this is the stage where all these things begin, prophase 1. During prophase 1, we can consider prophase 1 to have 5 various subphases. The first one we are going to be talking about is leptotene. This is considered to be the first stage of prophase 1. Please take note of something. I said the major difference between mitosis and meiosis is majorly within prophase 1, and that is what I'm about to explain to us now. During left-toting phase, what are the things that happen there? Remember, even during mitosis, we said there will be condensation of chromatid rather, at the level of our prophase 1, or at prophase, I mean. So the same thing applies here. During prophase 1 of meiosis 1, the first thing you have to have at the back of your mind is that there will be condensation of this chromatid to become more visible. During the interphase stage, that is the stage that precedes this prophase 1, it was not like that. The chromatid, they are not that visible, but during this prophase, they become more visible by condensation. So during left-toting stage, what happens is that there will be condensation of chromatid to become more visible. And also take note, at this particular stage, we already have sister chromatids. Sister chromatid. Sister chromatid are formed during interphase stage. That is when there is duplication of chromatid, the DNA structure of that particular chromosome. So that is what happens during the left-toting stage. Basically, condensation of the chromatid. The second thing is zygoting. During the zygoting stage of prophase 1, one of the things you have to have at the back of your mind is that we have what we call homologous chromosome. Homologous chromosome. Homologous chromosome. Homologous chromosome refers to the same chromosome having the same genetic constituent but different alleles. So when you are talking about homologous chromosome, it means a particular or a set of chromosomes that carries the same genetic information. For example, let's say this is one chromosome now, and this is another chromosome, and both of them are carrying information for, let's say, the coloration of the eye, the color of the eye. But I say they will have different alleles. So what I'm trying to say is that they have coloration of the eye, that is that genetic information they carry, but different alleles. So this one might carry blue coloration, while this one might carry, let's say, brown coloration. Blue and brown. So what I'm trying to say now is that we can consider these two chromosomes to be homologous chromosomes in the sense that they carry the same genetic information, that is, they carry information pertaining to eye color, but they have different alleles. The color they carry are different. That's what refers to the alleles. So during the zygoting stage now, homologous chromosome, remember when we are talking about meiosis, I said it occurs in sex cell. So during the zygoting stage now, homologous chromosome, one from the father and one from the mother, carrying the same genetic information, that is homologous chromosome, but different allele. Let's assume that the father is carrying, let's say, brown color for the eye, and the mother is carrying, let's say, black color for the eye. So the chromosome from the mother and the chromosome from the father, they will come together and align on the same side. Remember I said something earlier. We said sister chromatid has already been formed during the interphase stage. During the interphase stage, sister chromatid has been formed. So that means for each chromatid from both parents, or from each parent now, we have sister chromatid A, and also for the other parent, we have its own sister chromatid having the same DNA constituents. So at the end of the day, during the zygotic stage, one thing you should understand is that the major thing that happens during the zygotic stage is that these two homologous chromosomes, they come close to each other, they come together to form a structure we refer to as tetrad. That is having four structures. Tetrad. This is what we call tetrad. So that means left-toting stage, what happens there is condensation of chromatid. Zygotic stage, formation of tetrad. So the third thing now is a partitioning stage. During the partitioning stage, it is like one of the most important stages for prophase 1. For the partitioning stage, what will happen there is that there will now be crossing over of genetic constituents. What do I mean by crossing over? At any point in time when you hear crossing over, especially in human genetics, if you understand that we are talking about movement of genetic constituents from one chromosome to another, that is exchange of genetic constituents. So remember we said these homologous chromosomes have moved closely together now. They will now begin to exchange that genetic constituents. This to this, this to this now. Such that let's say we say the mother has the brown eye, right? The mother has the brown eye. Let's say this is for the mother, this is for the dad now. The mother and this is for the dad. The mother has the brown eye. The mother has brown eye and the dad has black eye. So let's use small letter B for black, capital letter B for brown. Capital letter B, small letter b. So this one will exchange genetic constituent A, this one will move A, such that the mother will later have what we call BB, whereas the dad will have what we call BB. This partition stage is very important because it allows for genetic variation. During this stage now, a new offspring may be formed, which may be or which will be a mixture of these two eye color now, such that this might totally form a new color of the eye for the offspring. Or in some cases, when we have dominant and recessive gene, the one which is dominant will be what the child, the offspring, will take after. So in the case, let's say the dominant one is this now, which is for brown eye color. Automatically, what will happen is that the child will have a brown eye color because the one which is black for the father is recessive. So during partition stage now, one of the things that will happen is that there will be crossing over, that is exchange of genetic constituents between the homologous chromosome, and as a result of that, it will allow for genetic diversity. That's what happens during the partition stage. The next stage is diplotin. During the diplotin stage, before I explain this, I want you to understand something. Just like we have centromere which join two sister chromatids together here, when tetrad is formed, there is something that joins them together, which we call chiasmata. We call it chiasmata. Chiasmata is the point of connection between two homologous chromosomes. And when tetrad is formed, even apart from the chiasmata, we have what we call the synapse. Synapse is a major form of communication between the homologous chromosome on the right and that of the left. So during the diplotin stage now, this synapse breaks down. But take note, the fact that I said synapse breakdown does not mean the homologous chromosome splits. So what I'm trying to say now is that although the synapse which serves as a connection between the two breaks down, but the chiasmata is still intact. As a result of that now, the homologous chromosome will still be tightly connected, but not as connected as before because of the synaptonemal complex that has been broken down. So that's what happened during the diplotin stage, that is this breaking down of the synapse. During the diakinesis stage, it's like the last stage of prophase 1. But I want you to understand something. Remember, leptotin stage, let me reverse everything I've said. Leptotin stage, condensation of chromatid. Zygotin stage, that is where there will be movement or moving together of homologous chromosome to form a tetrad. Partition stage, that is where there will be exchange of genetic constituents. Diplotin stage, that is where synapse will break down, so as to make sure the connection between the two homologous chromosomes are not as tightly bound together as before. And the last part, which is the fifth part, is diakinesis stage. For diakinesis is there to just make sure all the processes have been followed accordingly. And this is the stage where there will be total, full condensation of the chromatid in preparation for the next stage, which is metaphase. So diakinesis will make sure every activity here has taken place, and then it will also make sure the activity which takes place here, which is the condensation of the chromatid, is actually done in a right way. Diakinesis. So these are the stages of prophase 1, and these are everything that actually happens during the stage of prophase 1. So the next stage we'll be talking about now is metaphase 1. I want you to understand something. Remember again, when we are talking about mitosis, what happened during the metaphase stage is that there will be alignment of this chromatid on the center of the cell within the equator. The same thing applies here, but the difference is just that we say the homologous chromosome align on the center of the cell, such as we have a structure like this now. We have our spindle. So this is what we are going to get at the end of the day. That is, homologous chromosomes are aligned together within the center of the cell. Remember, they are still joined together by chiasmata. Chiasmata is still joining them together. Let's assume this is what we call the chiasmata now, trying to like hold the two in place. Chiasmata is still joining these two together, but the synapse has been broken down earlier in the previous stage now. So during the metaphase stage now, what happened is basically alignment of the homologous chromosome, not sister chromatid now, homologous chromosome on the center of the cell. So that's everything that concerns metaphase. For anaphase, so for anaphase now, what we'll be talking about under anaphase is similar to what happened under when we are talking about mitosis too. Remember, during anaphase, we usually say something. We say, if it is mitosis now, we say sister chromatid, the chromatid move to opposite pole of the cell due to the spindle fiber shortening. The same thing applies here. But in this anaphase stage now, one of the things I want to have at the back of your mind is that the chiasmata I've been talking about now, the chiasmata which I've been talking about earlier, will break down such that each homologous chromosome will go to the extreme end of this cell. That is, you have a cell like this. One homologous chromosome will be here, another one will be here, due to the fact that the chiasmata that combine them together has broken down, such that on each side of the pole now we have homologous chromosome for each genetic constituent now. So it is as simple as that. For anaphase stage, homologous chromosome breaks down and move to either side of the pole. And I want you to understand that each chromosome still has sister chromatid. There's nothing that changed during that particular phase, so it's still the same thing. So the next stage after anaphase 1 is till telophase 1. Telophase 1 is just preparation for cytokinesis. During the telophase 1 stage, everything that happened during telophase stage, talking about mitosis, still happens here. During the telophase 1 stage, the nuclear membrane will reform. The nuclear membrane will reform. Let's say this is the whole cytoplasm, one like this, another one like this. Nuclear membrane will reform on each, for each cell. This is what we call the nuclear membrane. The nuclear membrane, it will reform, and then making sure each daughter cell has its own nuclear membrane, so as for it to function effectively even after separation. So apart from the formation of nuclear membrane, I said that with preparation for cytokinesis, it is during this telophase stage that we consider as the pre-stage to cytokinesis. And by cytokinesis, I mean the division of the cytoplasm into two, so that we have something like this now, making sure each daughter cell will be split into two equal halves, having its own cytoplasm, so that the organelle there will be functioning for each cell separately. So that's what happened during the telophase 1, that is preparation for cytokinesis, formation of nuclear membrane, disappearance of the spindle fiber. All those things are what happens during the telophase 1 stage. And lastly, cytokinesis will occur. During cytokinesis stage, this will divide totally, and then we are going to have something like this. So we have cell A and we have cell B. Cell A, cell B. We have two daughter cells which are produced. Please, I want you to take note of something. During this meiosis 1 stage, haploid number of chromosomes are formed. Unlike during the mitosis stage, which we have diploid number of chromosomes, there is a reduction by half within the chromosome number for meiosis. So in the case of mitosis, we have 46 numbers of chromosomes, but in the case of meiosis, we have 23 number of chromosomes as a result of the process that has occurred during the cell divisional stage of meiosis. We have 23 number of chromosomes for meiosis, but for mitosis, just like in the previous video, we have 46 number of chromosomes. So that is how it works. It is now these two new daughter cells that will now go through the stage of what we call the meiosis 2. At the end of meiosis 1, we have two daughter cells. So these two daughter cells will now be the ones to go through the other stage, which we refer to as meiosis 2. Each of them will divide into two new identical daughter cells into two daughter cells which are not totally identical. Sorry for that. And then at the end of the day, we have four daughter cells. Four daughter cells will be produced at the end of the day. So I will now take you through the process of meiosis 2. For meiosis 2, it is actually identical to what happened during the mitotic phase. That is, everything that happens during the mitotic phase, talking about the prophase, metaphase, anaphase, telophase, is almost the same thing here. The only difference is just that we are dealing with haploid number of chromosomes for this case, or for the mitosis stage, we are dealing with diploid number of chromosomes. And also another difference is that we are dealing with sex cell here, but for mitosis, we are dealing with somatic cell. So majorly everything that happens during meiosis 2 is almost, or let me say, identical to what happens during mitosis. And for you to understand this, I made a video recently concerning mitosis. It will be nice of you to watch this video so as to understand every stage of this meiosis 2. But taking note of the fact that at the end of the day, we are going to have a haploid number of chromosomes, and we are going to have four daughter cells at the end of the day here, because we are talking about two daughter cells that enter meiosis 2. Based on what I described earlier, we have two daughter cells that enter meiosis 2, and then that two daughter cell will now also produce additional two daughter cells for each of these daughter cells, making four daughter cells. So that means for meiosis 2, at the end of the day, we are going to have four daughter cells. For mitosis, we are going to have two daughter cells. So basically, that's just the difference between mitosis and meiosis. So if you want to fully understand this meiosis 2 aspect, kindly go to my video talking about mitosis, and you'll be able to fully grasp the entirety of what meiosis 2 is all about. Thank you very much. Thank you very much for watching this video, and I hope this has helped you in clearing your doubts about anything that concerns meiosis, because it's actually a simple topic. So please do well to like, share, subscribe, and comment. And also, if you have any questions, kindly drop them in the comment section. I will answer them all. Thank you very much.